We're on. I'm not sure if there's so many people out there at the moment, so we're just going to give it a few minutes while we get everyone sort of signing up. But welcome to this Sophion webinar. We've got two great speakers today, Daniel Sauter, my colleague out in San Diego, and we've got a guest speaker, Dr. Mike Hendrickson, who's Director of Operations from BrainXell. Whilst we wait for, the slow coaches, those people that might be just grabbing themselves a cup of tea or coffee before they settle down for some wonderful talks, I'm just going to quickly show you a video of the Q-Patch Compact, which is our latest product to be released. So yeah, this is just a little bit of a teaser. Any more information, of course, just contact us. But while we wait for people to log in, and we'll get things going shortly, I'll just show you this video.
Experiment can be independent to investigate biophysical parameters, or they can be replicas to increase the statistical power of your experiments. In the Q-plate, where the cells are sealed and formed into whole cells, microfluidic flow channels and glass surfaces ensure good liquid handling. It is easy and ready to use when you are. Everything is explained on the screen and recorded for your documentation when making the analysis. Avoid the hassle and complexity of manual patch clamp setups. With Q-Patch Compact, you can focus on generating data. No hassle, just data. The data are stored on a separate hard drive inside the instrument.
I thought I would just cut that advertorial off, but yeah, that was just like a bit of a teaser to our Q-Patch Compact. We're pretty excited about it. So yeah, please do contact us if you've got any, questions or want further information on that. As I was saying, we will give it a minute or two, just to make sure that we get everyone who wants to signing in. But yeah, welcome everyone. This is our second Sophion webinar of 2022. We're doing a whole series actually in the next set of seminars on human-induced pluripotent stem cells, and particularly human-derived iPSC. Today's is all about neurons and glia for disease modeling and drug discovery. Later on today, Daniel Sauter, my colleague in San Diego, will be talking about some of the data that colleagues at Sophion, including Kadla, Daniel, and I believe May Zhang have created. Some really nice work, which is basically a collaboration that we've had with BrainXell on some of their excellent human-derived neurons and glia. But before Daniel speaks, Michael Hendrickson will be talking about their development of these human neurons and glia from iPSC, and how they've been used in disease modeling in neurodegenerative disorders on these cells. So I won't take too much more of your time, and I hope that we've given enough time for everyone to log in who wants to. And with that, I'll hand you over to Dr. Michael Hendrickson, Director of Operations of BrainXell. Over to you, Michael.
Thank you, Damien. And so I think everyone should be able to see my slides here. And good morning, good afternoon, good evening to everybody from wherever you're logging in. We've got people spread all over the world. So it's a pleasure to be here today to present a little bit about the iPSC field and in particular about iPSC-derived CNS cells. I'm keeping this very commercially neutral, so everything I say here is pretty much, applies to the field. I am an employee of BrainXell, obviously, but what I'm going to say is pretty much applies across the board. So this is me doing some science stuff. I was the first employee at BrainXell, and I'm currently the director of operations. I have a background in CNS drug development and neurodevelopment. And I would encourage anybody to reach out to me or to our group, whether you're a customer, potential customer, or never going to be a customer. We always really try to be as helpful as possible with any questions in the field. And so my contact information will be available at the end, or I'm on LinkedIn. And so feel free to reach out with any questions about the talk or the anything you hear today or the field in general, and we'll be as helpful as we can. So our overall rationale, the reason we started the company, is with a particular focus on drug development, but also in vitro CNS studies. And we know that it's a challenging and a time-consuming and a costly process, and it mostly fails, right? And I've been part of this myself. I mean, a lot of people logging on here have been part of CNS projects that are complicated and have a high attrition rate, especially once they get into the clinic. So our thesis is that you need to use systems that are as physiologically relevant as possible, because as possible that you can afford, or that you can make. And that's the reason we started the company, and it's the reason we provide the materials we do. And it's sort of what we strive to do, is provide materials that are as high quality and as representative as possible. Right? And so I'll talk a little bit about how we do that. And I'm going to talk a little bit about iPSC reprogramming, also neuronal and glial differentiation. And at the end, if we have time, I'll talk a little bit about using the cells in a general sense. And of course, Daniel will talk about using the cells on their systems. So everything that we do, and most things in the field these days, the human differentiations are being done from iPSCs, which are induced pluripotent stem cells, versus ESC, embryonic stem cells. They're very similar, the cell types. They're both pluripotent, so they can produce large number of tissues, and they're both self-renewing. But most of the field has migrated from ESCs to iPSCs. And so if you're not familiar with this cell type, they're made by taking some sort of biopsy, either blood or fibroblasts, from really any adult individual, actually any adult organism, right? So you can make these from monkeys or rats or mice. They're reprogrammed, that is, they're the differentiated cell, using the Yamanaka factors that can be introduced in any number of ways. They're reprogrammed to this iPSC cell. Those cells, very similar to embryonic stem cells, then are pluripotent, which means they can produce potentially, theoretically, any cell within the organism, and certainly it should be able to produce the 3 germ layers, right? Ectoderm, mesoderm, and endoderm. That's the definition of those cells. They can produce the 3 germ layers, and they can also recapitulate themselves. Like they can proliferate, right? Our concern, BrainXell is a super focused company. We just study the CNS. That's all we do, with the exception that we make one lineage that migrates into the CNS from the mesoderm, and that's microglia. So there's a little bit about, the reason people make these cells is because they can be turned, you can then, from an adult or an individual, with a control individual or an individual with a disease, you can generate, in theory, any cell in the body, right? And so that process involves differentiating the iPSCs into these various lineages and then into the cell type of interest, right? And obviously, our main focus is on neurons and glia. So that's the power of this system, right? Is to be able to take the adult cells from a human and then turn them into a cell type of interest to study in the dish. Our general approach is to take these cells and recapitulate development. So that's something, that's a key point here to think about. There are other ways to do this, but most companies and labs working in this area, or many, are doing a recapitulation of development, leading through these neuroepithelial progenitors into restricted neuronal progenitors, and then into early neurons. And this is just how we do the production, right? And this process takes several months. But the idea here is to take what's known about neuronal and glial development and recreate it in the dish, so that hopefully what you end up with is as representative as possible in vitro to the in vivo cell, right? That's the goal anyway. So this would be an example of making motor neurons. You're going to see some more data later from Daniel about are motor neurons that we may use on their system. But the idea here again is to take a patterning approach. So you're rostralizing, you're caudalizing, you're ventralizing, you're dorsalizing, and march down that pathway that occurs during development to make an ever more restricted cell type. And so in this case, these would be lower motor neurons or spinal motor neurons of the spinal cord. And these kinds of approaches then can be used, using different factors, different timing can be used to make a range of neurons and glia. So here are some images of the cells that we make, or others can make these same types of cells, and you verify we verify overall neuronal purity, and you verify also that the purity of the intended cell type, in this case cholinergic motor neurons, cat-positive motor neurons, are present within the sample, right? And the challenge in the field is making the material as pure as possible. It's always a challenge for us or other groups. But as I mentioned, a large number of cell types are possible. So, described in the literature, there may be 20 to 30 different types. From a commercial perspective, typically 7 or 8 different types are made. So we produce the spinal motor neurons and dopaminergic and GABAergic and cortical glutamatergic as well. And in our hands, we can make these at relatively large scale. And the goal is also obviously to make them very pure. In addition to making neurons, you can also make glia, as you might imagine, right? And so you can make a range of different types of astrocytes. We produce cortical and spinal astrocytes, and then we also produce microglia, keeping in mind that the microglia are from a different lineage. So that's a kind of analogous process, but quite a different production process in terms of what patterning, what factors are used to produce the microglia, say, versus a cell type of the CNS. So as I mentioned, one of the strengths of this technique is to be able to generate iPSCs from patients. And so in theory, this can be from any individual or any organism. And then you can produce the cell type of interest, or the cell type affected by the disease from that patient. So the idea here would be, if you look down at the examples there, to say, okay, someone has ALS versus because of this mutation, we can take fibroblasts from that individual, we can make iPSCs, we can make motor neurons, and in the end, we have human motor neurons from an ALS patient with a specific mutation. And that platform is potentially very useful as an in vitro system to do fundamental biology, but also obviously drug discovery and drug development. And these cells, these iPSCs, are available from many academic groups, and they're also, of course, available from biobanks. Please reach out if you want to find some and you're having trouble. We help groups do that all the time. There's a note here to remind me to say a word of caution about lines, because this is something we talk with groups all the time about. And if you remember actually anything from my talk today, please remember this, is that not all iPSC lines are created equal, and the quality of the lines can vary tremendously, even from biobanks, and the ability to make them into any one type can vary by line. So I think what we encounter sometimes with groups is they don't have direct experience in this area, so they read the literature, and in the literature it looks like, oh, iPSCs are all the same, they're all pluripotent, they're all self-renewing, there's no problem, one line is the same as the other. Our experience working with several hundred lines would say that's absolutely not true. And actually the quality can vary considerably. So that's just a word of caution. If you're thinking about getting into this area, or you're new to this kind of work, is to keep in mind that the quality can vary. The iPSC quality can vary from commercial sources tremendously. And the ability of any given line to make any specific cell type can also vary. So the way we get around that is to try to have some choices in what we do, and I test lines for various differentiation potential. So that's just a word of caution, I think, that helps. It's something that people don't publish in their papers, but people that work in the field for a long time are certainly aware of. So I wanted to provide some practical tips about working with the iPSCs. And this is I think useful if if you've never worked with them before, you're having, maybe thinking about getting into this area, and what's feasible and what's not feasible. So the cells that we produce or other commercial sources produce are typically frozen. Some organizations provide them, live cells, but typically they're in a frozen vial. And so the procedure is much like working with any other frozen vial, except that these are non-renewing cells, so they're going to be treated more like a consumable. And in that sense, you're going to thaw them and count them and plate them into the final, with the exception of what Sophion does, you're going to plate them into the final plate typically, right? And so they can be used across a range of different plate formats, from specialty plates like MEA plates, all the way up to very, low throughput 6-well plates, and kind of basically everything in between. The one piece of advice I have when people are starting to work with these cells for the first time is that that they're sensitive cells, and so versus say an indestructible cell line, so you need to be careful with what you do, and you need to be they can be somewhat finicky, so you need to take care in doing proper coating, proper washing, and obviously, media formulation is certainly important. Different from working with an immortalized line typically, because they'll be more sensitive to what to the manipulations you're doing with them. So this cell type, or iPSC-derived material always needs to be stored in ultra cold temperatures. You can't store them in -80 freezer, and they're very heat sensitive. So it's better to thaw them in a water bath that's 35 degrees than a water bath that's 40 degrees. And this, to some extent, this is commonsensical, but also, again, differs from many other cell types that people maybe have worked with in the past, particularly cell lines. And they're sensitive to osmotic shock, so we always tell people to do a very slow dilution to the cells and be gentle with them as you are diluting them up after thawing. And of course, obviously, it's important to do a proper count. One thing we work with people, or talk to people about all the time, is that typically, because these are terminally differentiated cells, they're pretty sensitive to the thaw process, and so viabilities in the 60s and 70s is pretty common, and maybe Daniel can speak to this as well. And so, I had a customer recently say, "Oh, we only got 75% viability." And I said, "That's okay. That's a pretty good number, actually," versus again what people are maybe seeing from cell lines that they've worked with in the past. And typically, what we're running assays, or many customers of ours are running assays in 96-well format, but you can certainly use 384 and on up. And the seeding is very important. People typically want to seed and not disturb the plate, and you can leave it out for 15 or 20 minutes. The cells are not particularly temperature sensitive, and so that's not an issue to get a nice even distribution of the cells. And then they can be cultured under standard culture conditions and using standard CO2 incubators, and they can be cultured for we've had cells in culture for up to 8 weeks potentially. The cultures tend to get a little bit more difficult to work with over time in terms of cells growing, or the neurite network growing on top of each other as time progresses, but you can take them out for that long. And so just as a summary of what I've talked about today, just kind of a few take-home messages. The iPSC-derived neurons and glia from human source is our opinion are the best best in vitro model system. They may not be the best overall model system, but the fact that the cells are human and the fact that the cells can be generated from a diseased individual are certainly strong cases in favor of utilizing this system versus a rodent originating neurons or glia and or a immortalized cell line. And actually, most of the groups we work with, people are coming from one of those two systems to introduce the human biology into their platform. And in general, they can be used with standard culture techniques and plasticware in various systems. There's nothing sort of special about what's needed there other than a little bit of extra caution. And they can be used on all sorts of different assays, again, with the idea that the cells are relatively fragile. So that has to be something that's kept in mind, right? And I think that's the last slide I have. And so, as I mentioned, we certainly, you can email me or connect with me, and we're happy to help with any questions in the field, whether people are customers or not. That's not really important to us. We're strong proponents of this kind of technology and working in the field and strengthening the field overall. And so that's my last slide. And I think we can do any questions that may have come up. I didn't really keep track, or we can give people a moment.
There are actually a couple of questions, Michael,
Okay.
and thanks for that talk. That was lovely and a really nice introduction. I think you made some really salient points there as well. I mean, things that, I don't know, people using cell lines may take for granted. They probably have to be a little more cautious, as you were suggesting, with some of these iPSC cell lines.
Yes.
But we do have a question. So Zai asks, are the spinal glia cells also generated by recapitulating the developmental process, which is the first part of the question? And then the second part is, what is the time frame for generating such cells? I don't know if you need those repeating.
No, I think those are really good questions. So just a little bit more background too. So there are techniques that don't recapitulate development to make various cell types, and those are sometimes called I might call them transdifferentiation, or they're sometimes called direct differentiation. And some of the attendees may be familiar with NGN2 methods. So that's a additional kind of reprogramming, taking a fibroblast or an iPSC and transdifferentiating it right to a neuron, or other cell type. Our approach within our company for all the cell types we make is really to try to, as closely as possible, recapitulate development. So the answer is yes. When you're making the glia, you use the same kind of approach as you would be for the neurons. The caveat there, and this is kind of interesting actually, is that any developmental neuroscientist here will remember that the neurogenesis precedes gliogenesis. And so the time frame required to produce the glia is often longer than the neurons, because that's somehow baked into the cells is that they have that timing mechanism, right? And we have a few tricks that we've developed to sort of push that process along, but typically a mature astrocyte will take longer to make than a mature neuron. In general, our protocols take from iPSC to the very end stage cell take about 3 months. And so they're relatively long protocols. Part of that involves expansion at different stages that are commercial aspects of the protocols that were introduced. But typically that's going from a nice iPSC culture to an early post-mitotic neuron or an early post-mitotic glial cell. The microglia are shorter. That process is about a month. Does that answer the question?
I hope so. Yeah, I think that was
okay.
a fairly thorough, and we've actually got a few more questions coming in, so I guess we probably should move on. So you was asking, can you comment on the utility of 2D versus 3D iPSC neurons in drug discovery? Essentially the current practices and maybe, future looking trends in those kinds of techniques.
So that's a good question. Current practices, at least from the groups we work with, our customers, our collaborators, it's mostly 2D. I think that's because the challenges of the readout in 3D, not necessarily the culture, generating the cultures. I think that's relatively well established, but the readout, whether it's imaging or other assays, there's more variability there. The readout is much more challenging than in a 2D system. So we do see though, more customers trying to add in the 3D, usually in combination with the 2D. And so I do see those systems coming in over time. I don't think it's going to happen as fast as you would guess if you went to a conference where everything's just 3D all the time, everything's organoids all over the place. Those are cool systems, but doing those readouts, especially in high throughput, is really challenging. So that's where we see the field at the moment.
Okay, and we've got another question from Radha. And I'll try and parse this for you as best I can. And if I'm getting this wrong, Rada, please speak up, because I may have misunderstood. But she's almost asking about a comment on neurogenesis, what people are working on, at present, and is such programming in terms of neurogenesis possible in human beings to, I presume?
I think I understand the question. Can you take the question maybe, can you take the existing neural stem cells we do have in our brains and promote neurogenesis? That's something that people are looking at. We don't have any, it's not something we've looked at or involved in. And my knowledge in this area is probably a decade out of place. But increasing in vivo neurogenesis in humans is absolutely a drug target for people. The best thing that can do that probably, though, is vigorous exercise, which is actually very well established to produce that effect. And I think that looks like, I can just read it myself here, I think, Damien. The next question looks like, are there any known markers of low quality iPSCs that cannot efficiently differentiate into neurons? That's a fantastic question. The answer is we don't think so, or we don't know. But it's a really good question. It gets to the issue I mentioned about iPSC quality, which is really stark, actually. I mean, I can't emphasize this enough. We get things that are absolutely beautiful out of the vial, and we get just stuff that's garbage, and it's unpredictable from where it's coming from, right? So, however, in general, if the iPSC quality is high, that means they're self-renewing and they're not spontaneously differentiating easily. So if that overall quality is high, typically the differentiation goes well. So we don't often see what we would consider overall high quality iPSCs have a failure to differentiate well. So it's the overall quality typically that leads to high quality differentiation. But for anybody out there that's thinking about investing a huge amount of money in a certain project that has a certain cell type they want to make, I would say test making the cell type first before you invest in editing a line or selecting lines or putting a lot of work up front. Run through the whole process first and confirm your line or set of lines or edited lines can make the cell type you want to make, because it's not easily predictable ahead of time. I mean, there's an excellent question, but the answer doesn't seem to be other than the overall quality.
Yeah, and I was actually going to ask something similar along those lines about quality control, because you did mention, obviously, one of your take-home messages is not all iPSC are created equal, and you definitely test for differentiation potential. Almost sort of following on from Randy's question, what kind of tests do you do now, and what kind of tests might you want to do potentially, to try and improve this idea of determining quality control of your iPSC-derived neurons and cultures that you've derived?
Yeah, that's a very good question. I mean, I don't think there's an easy answer to that question, other than, you could do a measurement, I think, of the level of spontaneous differentiation within a line, and then you could triage lines. The challenge there is sometimes lines, if you subclone them, some of the subclones can be much better performing than the overall culture. And so we sometimes work with lines a long time to clean them up. And the reality of working with these lines is very different from what you would get from what you might think if you just read published reports. But there isn't an easy marker to say, oh, that line will work, that line won't. Other than to say this overall quality is high or low.
Yeah, but I guess ultimately that's one of the kind of goals that you and other people in the field are almost working towards, is getting these very good sort of standard biomarkers, let's call them, that you might at some point be able to sort of say, well, it's got biomarker X in this proportion, which means we are fairly confident that this will be a very healthy cell line going forward type thing.
Yes, although I think those markers are going to be very rudimentary in the sense they'll be just what percentage of the cells are true iPSCs versus not. And I think with some of the newer techniques to establish lines, episomal reprogramming, the proportion of higher quality lines will be greater going forward. I think some of the newer techniques tend to produce higher quality lines. And also all this is unique to our laboratory, our handling, our protocols, right? So some lines that may work well for other people making hepatocytes may work very poorly for us and vice versa.
Yeah. Thanks, Michael. Actually, we've got a couple of questions now from you, but these are more pertinent actually to Daniel's talk, because I think he will actually potentially answer, or at least partially answer, some of you's questions around automated patch clamp and action potential recordings from neurons, for instance, or even characterizing iPSC neurons. So that was a really nice segue, that you provided us to actually lead into Daniel's talk. So Daniel, if you want to take over from here, and maybe you can address potentially some of you's questions, and I'll leave you to it.
Yeah, thank you very much. No, I'm excited to present some data today about a very fruitful collaboration that we had with BrainXell, and where we really sort of looked at these cells and characterized them on our 384 automated patch clamp platform. So I am going to address both of these questions throughout my presentation. So I'll leave it with that. And in case I forget it along the line, maybe you could bring it up at the end again. But so this data that I show, Mike did such a fantastic job to already set the stage and tell you about the relevance of using this technology and using iPSC-derived cells as a model cell. But I have prepared 3 additional slides that I just wanted to show here, because I want to provide some motivation why one wants to marry the automated patch clamp technology together with the iPSC-derived cell technology. And here I have this one review article that I've been reading some time ago from 2013 already, where the authors went ahead and looked at 842 small molecules and evaluated their success in the clinic. And then they looked at different parameters and tested whether these had a significant effect, a positive or a negative effect on the outcome in the clinic. So for example, here you could be looking at a publication per dollar spent in R&D, and then you would see that there's a significantly positive correlation with that fact to the success in the clinic. Or maybe not surprising, also the number of patents per dollar spent in R&D also significantly has a positive effect. But really what I want to highlight here with that slide is the therapeutic area that the authors have evaluated here. So if you were to set out to develop a drug against an infectious disease today, then you would have a significantly higher than average likelihood to be successful with that endeavor. On the other side, the only therapeutic area that has a negative correlation is neuroscience. And for that, Mike has introduced it already. There are several different reasons, one of which is that the clinical trials often don't have very well-defined endpoints, which just makes it much more difficult to evaluate these compounds. But furthermore, diseases are also often not as well understood. And then lastly, the model systems that are being employed or have traditionally been employed in the development cascades, they often don't translate that well into the clinic. So at this time, there was a need to take a step back and really reevaluate the way how neuroscience drug development is being done. And here in a review article from Ricardo Dolmesh, that nicely shows and evaluates these different models that are being employed in a cascade. So often we still see mouse models that are being used in such a cascade. And you see here there's a mechanistic insight versus throughput versus human relevance drawn out here. So the mouse models, they would be populating somewhat on the right left lower side or the right lower side, because it does allow you to gain some mechanistic insight, but the throughput is limited. And then it was shown time and time again that the human relevance is not very good. On the other extreme here, live human studies, of course, are very relevant, but the mechanistic insight is very limited and the throughput, of course, as well. So some 15 years ago, the iPSC-derived cell technology has entered the stage, and they really hold the promise to occupy that upper right corner here, where you could get that mechanistic insight. You can perform these high throughput screens and really test a lot of compounds on them and at different conditions. And at the same time, they hold the promise to provide a very high relevance in humans. So now I probably don't have to convince the audience here that ion channels are very important for various disease states, but just to highlight here, there are many of the important or devastating diseases that we still need to address better. They have ion channels that are involved in some stage in that disease progression. And to name a few, there's the Alzheimer's disease and Parkinson's disease, of course, but we also have spinal muscular atrophy, SMA, and amyotrophic lateral sclerosis. And the latter two, I'm going to dive in a little bit further, since we have received cells from BrainXell, which are some disease models for these two diseases. So if you set out to study ion channels, then the patch clamp remains the gold standard technique to do so. And I just want to remind the audience again why it really is that the patch clamp technique is so strong for such a study. So first, the technique allows to isolate individual current components in a cell, or in, for example, a native cell like an iPSC-derived cell, where there are a plethora of different ion channels involved. One can use the voltage clamp technique, as shown here on the top right insert, where you can use a voltage protocol and then evoke a current at a distinct voltage and really characterize this. It's also possible, since one can exchange external and internal solution, you can make a ionic substitution experiment and really zero in on a different type of ion channel. Importantly also, it's possible to use the technology for a biophysical characterization, as it's shown over here on the right top side. I don't think actually you see my cursor. One can really go ahead and do a mode of action analysis with that technology. You can see whether your compound has an effect on a different state of the ion channel, a closed and inactivated or an open state inhibitor. You can apply a compound from the intracellular side or the extracellular side to really dive into possible interaction sites with the ion channel. And there are many, many different other ways that you can use patch clamp to really interrogate the mode of action. Another important feature of the technology when talking about iPSC-derived cells is the possibility to get an absolute value for the resting membrane potential. And here you see in the right insert again an action potential recording. So it is possible to also determine the resting membrane potential of these cells. And often this has been used as a maturity marker or just a quality marker of a cell. So this is a very important property of the technology, and other techniques may not be, it may not be so readily available, this parameter. And then lastly, since it is a single cell technology, so you're recording from one cell at a time, there are different modalities where you look at several cells, but it's also possible to look at a single cell at a time. And because of this, it's possible to do a population analysis. You can really look at many different cells and then answer a question what the distribution, the variation in that population is with regard to a certain channel expression or with regards to several channel expressions. You've seen this slide already earlier, but I've copied it over just because I wanted to add some more words to it, how we've been using it. So all the work on the left side, as you can see here on the left side of that dashed line, was done in the lab of BrainXell. And then once they were cryopreserved and shipped over to our laboratory in Denmark, my colleague, Dr. Katla Røskva-Rosholm, who really done all the work, she's put them in culture here. And we're going to refer throughout the presentation to this point here at day 0 in vitro. So we then followed an SOP that was provided by BrainXell to further mature these cells before we then would put them on the automated patch clamp platform and record them. So that's how it looks. When we look first in the first row here, you have these micrographs showing the day 1 in vitro, 4 and day 7 in vitro. And as expected, we can see that formation of the network that is expected already at day 7 is a very nice network formation here. And if we then want to use these cells on our automated patch clamp platform, one requirement is to lift the cells into a single cell suspension. And really, that's the critical step. If you're working with both these technologies together, that's the step that typically requires most of optimization, because you just want to assure that you have this very clean single cell preparation where there's no debris or membrane fragments floating around that may prevent you from getting that giga-omic seal resistance. And so we've went through several iterations of assay development and then came up with that workflow as indicated here in the lower line. So after following the BrainXell SOP, we've added those cells into culture and waited different time points before we would then lift them using AccuMax, which in our hand really was the most successful approach, incubating them for, I think it was 7 minutes at 37 degrees. We then would use that cell suspension, spin it down, resuspend it in our external buffer, and then we would use often such a cell transfer plate where you have different chambers. And this is really a great feature of the technology because it allows you to do a head-to-head comparison between different cell lines. So you could add cells that were derived from a healthy donor in the first reservoir here, and then you would potentially add your disease model in the second one and add other ones in the others. But that allows you in one experiment to really address and do that comparative study all at once. After several iterations of assay development, we were quite excited to see a picture as it is shown here. So what is shown here is a 384 well plate view, and this is the result from the consumable, the Q-chip that you see here on the right side, where each square represents one neuron that is being recorded from. The green shading indicates that these wells passed our success criteria, which we now here set to 200 megaohm in resistance. And now I can just add sort of to what Mike had already introduced. So we were able, after some iterations of assay development, to get a 60% success rate. And this is a very satisfying, a very good result, I would say, since really, like working with a recombinant cell, which many people still use our technology for, or like initially most people would really only rely on recombinant cells. It's easy to get success rates in the 80s, 90s, yeah, in some cases even 100% in success rate. But the 60%, I would really look at it in that way, that 60% out of 384, that's still a tremendous amount of data that is being produced. And when we sort of compare it to the work that would be required to record it on a manual rig, then you can have, yeah, several, a large factor of time saving that can be obtained with that answer. Plus, you can make sure that you have that time-matched control. If you're really measuring two different populations, it can be a great advantage to do that head-to-head comparison. And I think that is the automated patch clamp is really an enabling technology for this. So once we had a satisfying success rate, we went ahead and used voltage clamp to characterize these cells. And when we just follow the first screen here, we use the voltage protocol as shown on the left top side here, where we stepped first to minus 120 millivolt and then went to minus 90 and had 10 millivolt increments to step up to plus 50 millivolts. You can then see the current response that is measured just after that step here. It's a very short time scale, which was then providing us with that characteristic transient inward current as one would expect it for a voltage-gated sodium channel that immediately inactivates here. So with our software, it's possible to extract the peak current automatically from this data and then plot it towards the applied voltage. And if you do so, you can construct such a characteristic current voltage relationship as shown here as the average from many cells. I forgot to mention that this first characterization was done on cells that were derived from healthy control donors. So this characteristic current voltage relationship, we were able to inhibit it by application of 1 micromolar of that peptide toxin, that tetrodotoxin from the blowfish. And this is as expected that these cells would have TTX sensitive currents available, and that was confirmed in that experiment. We also looked at voltage-gated potassium currents, and you can see in the left insert here, we recorded for most of the cells this A-type potassium current, but for completion, we also recorded in a subset of cells this more delayed rectifier type of potassium current as shown in the middle insert at the lower line. We also then went ahead and constructed the current voltage relationship that then had showed the expected rectification, the outward rectification, and application of a cocktail of 30 millimolar tetraethylammonium and 4 millimolar 4-aminopyridine, inhibit that almost completely here by 70% reduction at plus 60 millivolts. Now, I've mentioned in the intro earlier that the technology can also be used to do a population analysis, and that was exactly what we did here. We looked at the amount of successfully recorded cells that showed a voltage-gated potassium current. And in the top right insert here, you can see that this was pretty much for all the cells the case. And looking at the voltage-gated sodium current, we were able to detect it in 85% of the cells. And here we used 200 or minus 200 picoampere as a cutoff to identify one as a positive cell. So that was pretty good. In the next step, we also wanted to see whether these cells express any ligand-gated channel, and in particular, we used Gaba to evoke a current. Since sometimes some of the ligand-gated ion channels, they desensitized quite rapidly, and in a drug development approach, you might want to do consecutive stimulations. So you want to assure that once you applied your agonist, that you can completely wash it out and then apply it at a second time again, a third time, potentially including a compound to test for an effect. But to do that successfully, it's important to keep that ligand exposure time to a short time interval. And because of this, we have developed a stacked solution approach that is sort of represented in this comic on the left side, where we first pick up a wash solution, external saline solution, and then we pick up in the lower compartment here the agonist containing solution. You will then have some mixed phase that is sort of indicated in the green, but we have validated that over and over again to show that really this mixed phase is small, and you can conduct these experiments such that you, now looking at the current response, you add the agonist, you get that sharp increase in the current, that inward Gaba current, and then you see a inactivation of that channel or desensitization, and then you wash it out and you completely come back to this original baseline. We were able to detect Gaba mediated current in 45% of the cells. So in the next step, we wanted to look really at, still in these cells derived from a healthy donor, we wanted to look at the time in culture. So you see in this plot here on the x-axis, there are different time points, 5, 11, and 15 days in culture. And then we looked in an absolute fashion at the various different current components that I've just introduced. So the longer we kept these cells in culture, I hope you can appreciate from this plot here, we've seen an increase in the functional expression of these ion channels. Although looking at the black bar for the potassium channels, we've seen that it leveled off already at day 11. So there's a benefit in keeping them slightly longer in culture. Well, okay, that was unfortunate. There was an animation here on the right side, and that should have disappeared. And Jane gave me a warning that this has been transferred in a picture, but unfortunately I missed that one. So I'm describing with words that what happened here now is that unfortunately we have an inverse relationship with the success rate, such that the longer we keep those cells in culture, we see a decrease in the success, unfortunately. So the numbers were 60% routinely that we would obtain at day 5 or day 7 in vitro, but then going to day 11 and day 15, that dropped to sort of 30 to 40% in success rate. And that is probably attributed to the fact that you have this network formation. And once you have that network formation, it just becomes more difficult to get that pure, clean single cell suspension, since you need to disrupt these cells in a more vigorous way. So I mentioned we were lucky to have these disease models at hand. And the first one I'm going to talk about is a cell line that we received from BrainXell that was derived from a patient exhibiting spinal muscular atrophy. So SMA, that's a very devastating disease that is often fatal if it's an infantile onset, but it is in 95% of the patients caused by a mutation in the SMN1 gene, and that through several steps then leads to a degeneration of the lower motor neurons, and it can lead to paralysis or several very bad adverse effects for these patients. So there's an unmet need. So fortunately, Roche and PTC Therapeutics, in combination with SMA Foundation, have set out to develop a small molecule drug that's shown here on the left side, the SMNC3, that is now approved by the FDA as, I believe, the only orally available treatment. And we were also lucky to have this compound at hand when we evaluated these compounds, these cells. So since there's some hyperexcitability in the motor neurons, our scientists focused in really on the voltage-gated sodium channel and looked if there was any difference. So if we look first at the black line in the top left insert, looking at the cells derived from a healthy donor, we were able to construct that current voltage relationship for the sodium channel. Comparing it with the SMA patient-derived cells, we've seen a significant increase in the current amplitude. And we looked over on the right side if that is potentially due to a change in the gating of the channel, and therefore constructed this activation curve as shown here. But here we didn't detect any difference. So that would indicate that the increase in current is likely due to an increased density of these channels in the plasma membrane. We then used that SMNC3 compound and pre-incubated the cells for a day. And we were then quite happy to see that this lowered the amplitude to the same level as we've recorded them in the control scenario. And again, there were no changes in the activation kinetics of the cells. So we at Sophion, we strongly believe that there's a change in paradigm, that automated patch clamp is sort of becoming the new standard, the new gold standard. But nevertheless, it's always nice to sometimes make a comparison to manual patch clamp data. And here in this particular case, there was a study in Science from Liu et al. From 2014, and the group performed the exact same experiment as we done them on automated patch clamp. And it's quite nice to see that the results they've obtained are exactly in line with what we found on the automated patch clamp system. So we've tested another disease model, and that was derived from a patient exhibiting amyotrophic lateral sclerosis. And you know probably that disease from the late Stephen Hawking, who unfortunately passed away. He was suffering from that disease early on already in his life. But the disease also gained some attention in the social media during that fundraiser, the ALS Ice Bucket Challenge, and that was quite popular at the time. You can see here Bill Gates on a picture who is just getting some water poured on top of him. So that disease is in 20% of the familial ALS is caused by a mutation in the cyanide zinc superoxide dismutase, the SOD1. And then following several steps, it also leads to a degeneration and death of the motor neurons. In a similar fashion as with the other cell lines, since there's a known hyperexcitability of these cells, we used here that cell transfer plate that I've introduced at the beginning to do a head-to-head comparison. And here we didn't have any small molecule or any pharmacology available, but instead we received a isogenic control where this mutation in the SOD1 gene was corrected again. And you can see that here in the lower part. So first, following the black line for the control cells, we were quite surprised about how drastic the effect in the increase in that current amplitude is here in this ALS patient-derived cells. And then the isogenic control again really reduced it back to these original levels as shown in the control level. So that was a quite nice result. With this, I only have one slide left, and that is, I hope that I was able to summarize or to convince you that there has been tremendous progress with the iPSC-derived cells on automated patch clamp, and we do believe that this technology has now sort of matured to a state where this can be readily used for a drug development process. We are excited to see sort of what the field will use this technology for. We get a lot of requests already from our customers, and there's interest in this. And we see that there are possibilities for disease modeling. It can be incorporated in drug development processes, but there's also a big chance for precision medicine, we find. And now, since the question was already there from you, I'm potentially already jumping to my backup slide. Oh, no, there's one in between. Of course, most importantly, I have to acknowledge my colleague that I mentioned, Dr. Katla Röskva-Rosholm, who's really done all that work in that study. And then also BrainXell for providing us with these great cells, but also with the support throughout the study. So I think that was a really great collaboration, a lot of fun to work on. But what I mentioned with my backup slide, because there was the question regarding current clamp. So it is absolutely possible to do current clamp recordings on the automated patch clamp platform. Of course, the criteria of success, the quality needs to be much, much higher. We have recorded an action potential as it's shown here in this figure. However, I haven't included that in the main presentation since we haven't recorded all too many of them. So there might potentially be some optimization needed to really use this routinely. But I think as a proof of principle, this already is a quite convincing data that we can show here. And I think we already have some ideas how to further improve this, and it would just need to go through some assay development iterations where we really zero in on the current clamp. Right, I think with that, I am going to jump back to my summary slide and take any questions if there are any.
Talk, Daniel, thank you. And as you rightly noted, it's some beautiful data from our brilliant colleague Katla Röskva-Rosholm. So, you know, all the thanks and plaudits should be going to her.
Yeah,
we're the lucky ones that get to present this beautiful data and pretend in some way that we've had some sort of hand in it, but it's actually no, it's all the brilliant people you know, at Ballarup and particularly Katla who's made this beautiful sort of data possible. We've got a number of questions, Daniel. You've obviously done a great job of answering news that sort of came into us almost at the halfway point and segue into yours. I should mention actually, which I've done in the chat, but I'll just reiterate, I believe Michael Hendrickson's probably left the building by now. So if you do have any questions specifically for him, do get in contact with him, or we can put you in contact, or we can pass on your questions. But whilst we've got Daniel here, we'll go into some of the questions that have been coming in for Daniel. So Zai was asking, what is the latest time point, i.e. the days in vitro, one can successfully dissociate motor neurons? That might be more for Michael, to be honest, but you can have a go at it if you can.
Yeah, so I mean, we have tested, the latest point that we have tested was the 15 days in vitro. At that point, we've seen, I mean, we haven't shown the micrographs for this, but I mean, there was a clear network formation, and we were still able to get a single cell suspension there. Although, I mean, there was that missing plot that I unfortunately can't show, but I told you with words that we were still able in that scenario to get a 30% in success rate. And then just to remind you, with a 384 platform, that's still somewhere about 120, 130 cells to look at. So if that's really sort of the time point that you feel confident about, that this is how far you want to go, then I think that is still something that you could be doing with that technology.
And on a similar line, Charles was asking, how long did you have to differentiate the cells to get electrophysiologically mature neurons? Yeah, so I think it's probably going to be sort of an averaged answer. I mean, like some of them would, of course, be you detect mature cells that are firing action potentials already early on. There might be a benefit of waiting longer. The action potential that I've shown you here was recorded at day 7 in vitro. So there might be a benefit, or there's likely a benefit, what you've seen sort of from the expression pattern, the functional expression pattern, that you wait a little bit longer, but you're probably just sort of shifting the average. I think you probably get already good recordings after day 7. Okay, and on to the next question. You was asking, what's the percentage of neurons in the cell suspension? For instance, from the last talk, he was saying often that the culture is mixed with glia and neuron. So what kind of percentage of neurons in the cell suspension are you getting from a potentially mixed population in culture or from culture? Yeah, it's a great question. So I mean, we can only judge from the electrophysiological signature that we are reading out, where we then can differentiate with expected patterns. I've mentioned sort of this when I talked about the voltage-gated potassium channels, that we've seen those that expressed the A type. Those were typically also the ones that then showed a transient inward current, which really would resemble a more mature neuron type, where the ones with the delayed rectifier type, that maybe more of another cell type. But here, after this process of lifting, although I don't have like very hard numbers where we've looked really at that ratio with a statistics, it was the vast majority of the cells that we have successfully recorded, meaning they had the capacitance and a resistance threshold exceeded. Those ones, I would need to look it up in detail, but it was somewhere in the 75-80% range of the successfully recorded ones. And presumably most of the other cells would have been glia, or would they be other potential, I don't know, cell types? I would, I mean, there might be progenitor cells, but glia cells, yes, is probably something. I think that might also be a follow-up for Mike, who has a more precise answer to that. Okay. And Kalai was asking a two-part question. Does the capacitance of the cells increase when cultured for more days than corresponds to the decrease in the success rate? And does culturing for more days favor the maturation of these cells? So the capacitance was stable. That's why we didn't show the densities here, especially when doing the comparison in the disease models. Also, so we had the capacitance was stayed, and then for more or less favor. Yeah, and then absolutely. I mean, what we found is that keeping them longer in culture, that certainly benefits the maturation of the cells, since we are also using that proprietary process that BrainXell had provided us, which further matured the cells even when we had them in culture in our incubator. Okay, and We have another question, which might be the last one. So if you've got a burning question, get it in soon, because this might be the last of the go. So, of course, being the last question, it's bound to be the best question. So this goes to Hi. There seems to be a shift in the voltage dependence of the nav currents going from the automated patch to manual. If that is correct, can you comment on why this might be?
Yes, absolutely. And that is a really great question. And it's good to have really that detailed look at the different data. So what we often find, or what is a well-known fact, is that if you're using a solution that includes fluoride in the internal solution, then this may have an effect on the gating of the voltage-gated sodium channel, such that it shifts the activation, but also the inactivation kinetics. So for this particular assay, there was some fluoride included to help foster the seal formation of the recording. But so now it's important to say, since all that work that I've presented today was done on our 384 well platform, the Cube, which you can see here on that last slide as the black machine. But right next to it is the QPatch technology. And that QPatch line of products that we have now is a technology that allows you to also obtain giga-ohmic seal resistances without the use of any seal enhancer, including fluoride. So we also have done some experiments on the QPatch, and here you typically see that this is not an issue. So we typically really recommend, if you're looking at looking at mechanistic studies, the QPatch might be the better choice. We found that with the Cube, many people still use it for voltage-gated sodium channels, since you can do this time matching. So it might not be so much of an issue if you're testing a compound, but you're testing all the compounds at the same time in the experiment. So you know that any sort of shift in the gating kinetics that may have occurred would be sort of time matched and at the same time, which then allows you to rank your compound equally. But this might be the reason for this change.
Great. I think, unless someone gets a very quick question in soon, that was the last question. So lovely talk, Daniel. We've actually got one just come in, just popping in last minute. Look at this, Arturo gets in right at the death. In respect to Hi's question about shifting the voltage dependence of sodium currents, I myself have had that shift in my own results with APC instruments. It might be due to the solutions of different ionic strength, he suggests.
That's a good point as well. That's absolutely absolutely a good point as well. I mean, of course, if you have sort of a different driving force, you could imagine, although this was a normalized, so it was a normalized current, so G over G max, but certainly, I mean, the composition of the solution, that always is sort of the first point to look at. And I think there are different approaches to look into. I think like we could also sort of really zero in on what kind of series resistance the group has had. And I think here for this studies, we've really included only the ones that had sort of a low series resistance, which certainly also helps to get a clean readout.
Okay, and I think that might be the end, and you probably need to sort of get on with your working day, and the people in Europe probably need to go off and get some dinner. I'll certainly be doing that shortly, but thanks, Daniel. That was a lovely talk, and thanks obviously to Michael Hendrickson. I believe he's taken off, so again, if you do have any questions for him, please get in contact with him directly, or we can certainly put you in contact if you've got further questions about some of their great iPSC-derived neurons. Thanks, everyone. Thanks for your attention. And, of course, if anyone has got further questions for us or for Michael, it's only an email away. Just get in contact with us. We'll do all we can to answer any questions on these and any other burning ion channel issues. I'm probably bigging it up a little bit too and making it far too dramatic. But anyway, you know what we're saying. Thanks, everyone. Thanks, Daniel. Thanks, everyone in the background as well. People like Jane, who do a lot of organizing and arranging for these webinars. They often don't get the thanks and the plaudits that Daniel and I get because we're up here up front. But yes, thanks to everyone who's made this happen, and thank you all for your attention. There's Jane right now. You can take the applause.
Well, applause to the speakers and to you, to yourself. So it was great seeing so many. But have a great day, evening for my sake. So see you.
Take care, everyone.
Thanks,
Bye.
everyone.